Okay, so let us start with the third video of this series, which is mechanism of evolution. So, moving on with that. So, let us first start with the basic of Darwin's theory of evolution. So, like I have already talked about Darwinism, so Darwin's theory of evolution was based on the fact that it is very slow and it slowly progresses towards adopting the best practices and innovating some things which are very useful, and then trying to be the industry leader, that is survival of the fittest. So this theory of evolution was given by Darwin, which is like always ready for the competition. So human beings, they have competed with among each other and also among different species, and then they have evolved to be one of the best species. So that's why they are quite ahead of the other species, ahead than the other species, and therefore this kind of evolution is given by Darwin's theory of evolution. So Darwin's finches in Galapagos Islands. So Darwin observed in Galapagos that the finches, like these birds, they have different beaks, different beak adaptability, and this rapid diversification of this one species into several different species is mainly because of their different feeding habits. So if you can see here that some are fruit eaters, some are insect eaters, some are cactus eaters, and some are seed eaters, and according to that their beaks have modified and adapted to the type of food that they are eating, like parrot-like bill, grasping bill, probing bill, crushing bill. So these bills or beaks have developed because so that in order to reduce the competition among themselves. So that's why these finches, they occupy different niches in the same habitat, that is Galapagos Islands, and therefore since their niches are different, that means their food habits are different, and so they do not compete with each other, and there is less competition for the resources available in those islands. So this kind of adaptability is known as adaptive radiation, which means that due to this adaptive radiation, one single species develops into different alternative forms in order to reduce competition and in order to make sure that they are surviving at their best of their ability. So this another example is given here of Australian marsupials in Australia, and they have evolved from a common ancestor, and they have evolved into these several types like tiger cat, marsupial rat, kangaroo, bandicoot, koala. So all these different types of marsupials have evolved from a common ancestor, just because they do not compete among themselves and they share different niches in the same habitat also, and therefore there is much less competition, much less fighting among themselves. So this is known as adaptive radiation, and in order to do that, you have to first understand what are the different types of evolution. So first of all, what is the divergent evolution? Divergent evolution means that when an ancestor gives rise to 2 or 3 different types of species, so closely related species gradually become more and more dissimilar. So from one common ancestor, there are 3 or 4 different species, and they are all dissimilar and they are closely related, but they are very different from each other. So next is the convergent evolution. So in convergent evolution, that means that species from different evolutionary branches, they resemble each other and they have similar ecological roles, which means that they have similar features. Like you can see this reptile, it's a reptile, it's extinct, but like this is a fish, shark, and this is a mammal, dolphin, and this is a bird, penguin. They all have similar features among themselves. So that, but they are all from different ancestors, because it's a mammal, it's a fish, and it's a bird. They have different lineages but still they look almost similar. They have several features which are common to each other, and the kind of evolution is known as convergent evolution, where they are not coming from a common ancestor but still they are very similar. And here the ancestor is same but still they are very different, so that is just the reverse. So this is called convergent evolution, and this when a common ancestor but two different species, then this is known as divergent evolution. Next is the parallel evolution, where two related species arise from a common ancestor and two species then evolve in the same way over time, and then the same kind of environmental pressures are upon them and therefore they adapt in the same way throughout time. So the like example is woolly mammoth and modern elephants. So woolly mammoth is already extinct, but from a common ancestor these two developed, and elephant has adapted itself in a different way and mammoth adapted itself in a different way and it became extinct. So this is an example of a parallel evolution, where two related species arise from a common ancestor and they evolve in two different in the same way but separately. So this is known as parallel evolution. Homologous structures are same structures but with different function. I will come to that later. And vestigial structures are structures not presently used but they had an ancestral function, like we have our tailbone, appendix. These are vestigial structures which we are not using anymore, but still they are there because of the evolution, and they had ancestral functions. So this kind of parallel evolution is also seen in bees, wasps and ants because they have developed social behavior. These are known as social insects. So these insects also show similar behavior and they have evolved similarly, but they are two different species and their lineages are completely different. So this is parallel evolution. So like I was saying, this divergent evolution is when from the wolf, which is a common ancestor, you will get fox and domestic dog. But the convergent evolution is no common ancestor, but still there is a bird with a wing, a butterfly with a wing and a bat with a wing. They all have similar features, they all look similar, but still they are not from a common ancestor. So convergent evolution decreases the morphological differences between species, and the species to species differences just become less, and also unrelated species become more and more similar. So they appear similar to each other, they look like each other. In divergent evolution, they increases the differences or between the species. They become more and more different or more and more dissimilar. So this is the difference between divergent and convergent evolution. And parallel evolution, two species evolve independently of each other, maintaining the same level of similarity. We also see that in some cases of leaves of plants, where the plants have clearly evolved differently, separately, but still we can see that in several plants with distinct leaf forms that are evolved in parallel. So convergent evolution and divergent evolution, different ancestor, common ancestor, they converge to produce analogous structures and they diverge to produce homologous structures. Species appearance becomes more similar over time, appearance becomes more different over time, and species are unrelated, they are genetically different, and species are closely related, they share genetic homology because they are coming from a common ancestor. And example is the pentadactyl limb structure in vertebrates, and this example is the wings in insects, birds and bats. So what about the pentadactyl structure, limb structure in vertebrates? So if you see here that the limb structure in frog, lizard, human and bird, they are all same, but they externally appear to be very, very different from each other. So they differ morphologically. This is known as homologous organs. And analogous organs meaning when they look externally, the look is very similar, but still they are structurally very different from each other. This is known as analogous organ. So homologous, they differ morphologically, and analogous, they show superficial resemblance. Homologous, they have internal structure same, and analogous, they have internal structure quite different. Like I was saying, that an insect wing and a bird wing are both very, very different. The homologous organ, they develop in related organisms, and analogous organ, they develop in unrelated organisms. So stages in development are similar, they have similar developmental pattern, and they have dissimilar developmental pattern, because an insect wing is feathery or rubbery, but a bird wing is made up of bones, flesh and feathers. So they are all very, very different from each other. So they have similar functions because the wings in both cases, they help to fly. But in case of homologous organs, they have different functions, because for a bird they help to fly, for a human it's like for any purpose of the hand. Lizards, it's for like crawling, and frog, it's for jumping and all that. So they have different functions, and they are inherited from a common ancestor, but these are not, and the examples are quite clear, I guess. So these are homologous and analogous organs. Another type of evolution is called co-evolution, where species adapt together. Like when an example is given here, when the hummingbird and the flower. So when the flower is evolving and the hummingbird is also evolving in order to adapt to its changes. So the hummingbird's bill is long enough to access the nectar source of the flower, and therefore the flower is also benefited because it is getting a pollinator, and the bird is useful for its pollination as well. So the bird is deriving its nutrition and also helping in its pollination. So in that way, both are helping each other, they are adapting each other, and so when the flowers have evolved such that their nectar becomes hidden, then the hummingbird's bill or beak also evolved such that it is very long and slender and can go inside and take out the nectar. So this kind of evolution, where both the species who have an interrelationship among them, both the species are evolving together, this is known as co-evolution. Now coming to speciation. So speciation means the formation of two or more subspecies from one particular species. So this happens due to several reasons. So the first and foremost is whenever there is a barrier or a geographical barrier in between them and they get separated physically, then this kind of speciation is known as allopatric speciation. Whenever there is a spatial isolation among them, so that means that in case of parapatric, there is a new niche here, but there is a partial overlapping of these two niches, then this kind of speciation is known as parapatric speciation. When part of a population becomes separated, goes into a new habitat and starts living over there. So, but still it is not completely separated, there is not any barrier between them, then we can call it a parapatric isolation or speciation. In case of a sympatric speciation, it occurs in populations living in the same geographical area, where what happens is that when there is a genetic polymorphism involved, then it is known as a sympatric speciation, because the genotype of some organisms become different, and therefore they have to form a new species in order to survive. So this kind of speciation is known as sympatric speciation, where there is a genetic polymorphism within the population and they start living separately. Next is the peripatric speciation. isolation of the a population at the periphery. Like there is a, from the periphery it just pinches off and goes to somewhere, live in somewhere else, and this is known as a peripatric speciation. So allopatric speciation is very common because it is a geographical speciation. It can happen whenever there is a mountain range between them, or a river, or a water body, large water body like an ocean or something, and then they get separated and therefore the speciation occurs. Peripatric is whenever there is a mainland and an island population. So the island, when it gets separated, it is like from the periphery of the main population, and also it happens because there is a genetic drift, therefore it forms a different species. And parapatric speciation is when a small subpopulation remains within the habitat of an original population but it enters a different niche. Then it is called a parapatric speciation. And speciation is said to be complete when they are not breeding with each other. So interbreeding is not happening and only they breed with the members of their own species. Then we call it the speciation is complete. And sympatric speciation is involving genetic changes, genetic variations, and therefore it is quite rare in nature. Mostly the other types of speciation occurs quite commonly. So divergent evolution leads to speciation, and speciation can be of all these types. So like I was saying, what is genetic drift? Genetic drift is the change in allele frequency due to a chance event. So I have already discussed about alleles and when I was discussing Mendelian genetics. So if you can remember, there are two copies of each particular character or trait in both the chromosomes. So if the allele frequency, so if we consider that there is a two kinds of allele, the red and the blue, and the ratio is equal. So something happens, like a very abrupt event happens, and then half of the population goes out, half of the blue goes out, and it becomes 1 blue and 5 red. So the ratio after reproduction will be obviously 10/2. So this unequal ratio will remain increasing generation after generation, and this is known as genetic drift. So we have started with 10 alleles, but some random sampling or something chance event has occurred, and then 6 of the original alleles have left descendants. Several generations of random sampling includes that only 2 of their original alleles and their descendants are left, and the rest have just gone. So this kind of drift in the genetic composition of a population is known as genetic drift. And this genetic drift is chance event, and it is not natural selection. It changes the genetic composition of a population by chance. They are random events that changing the allele frequencies over time. They reduce the amount of genetic variation in the population. So first there were 10 alleles, now we have only 2 alleles. So ultimately this blue ones will all be eliminated, and only the red ones will be there. So this will decrease the number of alleles or variation in genetic variation in the population, which was there once but it is now not now. So it can cause fixation of one allele, that is one allele occurring over and over. Like you can see here, the red ones have increased. So two main types of genetic drifts can occur. One is the founder effect and the other is the bottleneck effect. So founder effect is, like I was saying, that a few individuals from a population, they start a new population with a different allele frequency than the original population. So since suppose the original population was like this, and then few individuals, the blue ones have separated, the orange ones have separated, and some orange and blue have also separated. So these are founder populations and this is the original population. So A new colony is formed by a very small number of individuals, and they are called founders from a larger population. So this happens whenever there is an island formation. So this is the mainland, and only this red type just drifted away and went to an island. So it will increase the population of red insects only. So therefore the island will have only red insects, and the mainland will have a combination of all types of insects which were already there in the original population. So the smaller the colony, the less its genetic makeup will be there, the less genetic variation will be there. So depending on the success of the random drift, this will continue to affect the frequency of the alleles and contributed to the evolutionary divergence of the finches. So Darwin's finches is also another example that arrived as strays on the island, and then they started developing their own modifications so that they can survive with each other. So a new population whose gene pool is different from the original parent population is known as genetic drift, and it is the formation of a separate colony which is known as a founder population, and this kind of phenomena is known as founder effect. Next is the bottleneck effect. So this is a dramatic loss of genetic variation due to some environmental effect on our human activities. So examples include fires, droughts, overhunting, earthquakes, destroying habits. So genetic drift with bottleneck effect happens whenever there is a chance event which greatly reduces the size of the population. Like the population size has decreased like this because of some event which has happened, and therefore the rest of the population are trying to recover the population size. Now this becomes a bit tricky because original population, suppose it composed of red and blue genetic members. Now, when there is a bottleneck event happening which reduces almost a majority of the population, then only a few red individuals survive and they are passed to the next generation, so they can pass their genes to the new red population. So in the next generation, if you see, there will be an unequal ratio of the alleles or the genes present rather than in the parent population. Because so here also the new population shows a strong dominance of red over yellow alleles, because the surviving individuals have allele frequencies different from that of the parent population. So this kind of phenomena is known as a bottleneck effect. So founder effect versus bottleneck effect. Like I was saying, that this founder effect is a phenomena when a small group of individuals becomes isolated from a large population. But bottleneck event is when a population rapidly decreases in size. So this can be caused by migration, and this is caused by natural events like droughts, floods, earthquakes, etc. And it does not affect the original population, founder effect, but this bottleneck effect affects the original population, and therefore the population, it produces a population of non-random sample of genes, and this produces a population of random sampling of genes from the original population. So this is the difference between founder effect and bottleneck effect. Next is gene flow. So what is gene flow? Gene flow is basically the movement of genes into or out of a population caused by immigration, that is migration of individuals into the population, or emigration, that is migration of individuals out from the population. So population may gain allele or lose allele depending on immigration or emigration, and this gene flow reduces the differences between the populations because there is an inward or outward flow of alleles, and therefore the difference between two populations will decrease if there is a constant flow of gene or alleles between them. So this is known as gene pool, where there is a barrier, gene flow where there is a barrier, then we have no gene flow. But whenever there is no barrier and migration can happen from this population to this population and this to this, then you can see that there is unrestricted gene flow occurring over here, and therefore there will be less and less dissimilarity between the populations. So What is the difference between gene flow and genetic drift? Gene flow is alteration in gene frequency due to migration, and genetic drift is pure chance, like how the randomly it will separate from the original population we don't know, or if a bottleneck event happened, that also we don't know. So gene flow occurs due to exchange of the genes in the adjacent populations and through interbreeding. But genetic drift occurs by accidental or sudden elimination of a particular gene by founder effect or by bottleneck effect. And larger populations tend to show more migrations, therefore there is a more gene flow. And genetic drift, smaller populations have greater chances of genetic drift because they have a tendency to just segregate from the original population. So what is a gene pool? A gene pool is a collection of all the genes and all the alternative forms of genes, that is all different alleles present in any population of a particular species. So this can be any population, like the frogs in a pond, all of their genes together. For example, if you have a population of cows of different colors, then the colors will be of meaning they have different genotypes also. So that all the genotypes, all the different allele combinations taken together, if we consider it as a total pool, then it can be called as a gene pool. Next is the Hardy-Weinberg principle. So Hardy-Weinberg principle is a very known equilibrium equation that was given by Hardy-Weinberg. So they try to understand that how can we calculate the allele frequencies in a given population. So if these 7 conditions are satisfied or fulfilled, like if no mutation is occurring, if no natural selection is occurring, if the population is very, very large, all members can breed, and that is a totally random breeding, everyone produces same number of offspring, and there is no migration in or out of the population. If all these conditions, 7 conditions are fulfilled, then we can calculate the allele frequencies. But we know that in nature, these 7 conditions can never be fulfilled, and it is not possible to create a population with all these criteria. So it is highly unlikely, so it will happen in the real world, but still, so there is no such practical application of this equation, but still in population genetics it is a very important part, so you have to go through this. So Hardy and Weinberg, they developed a simple equation to probable genotype frequencies in a population. How are the allele frequencies different, or what kind of genotype frequencies are there in a population, that can be known by this Hardy-Weinberg equilibrium equation. So if you remember the Mendelian genetics thing, that there is a dominant allele which is always expressed whenever it is there, either in heterozygote or in homozygous, like even if it has a 2 copy or it has a 1 copy, the dominant allele will always be expressed, and there is a recessive allele which will be suppressed. So if these 2 alleles are denoted by capital A and small a, then P is the frequency in which the dominant allele is expressed in the population, and Q is the frequency in which the recessive allele is present in the population. So these 2 taken together, if P equals all the alleles who are homozygous dominant and half of the alleles who are heterozygous. So capital A small a will have capital A, 50% of heterozygous alleles will be capital A, and 100% of homozygous alleles will be capital A. So these 2 together equals to P, and the same way it goes for Q. So P is equal to capital A plus half of capital A small a, that is capital A homozygous plus half of heterozygous. Similar thing goes for Q, which is for the recessive allele, so half of heterozygous and recessive homozygous. So in this 2, the P and Q should together make up 1, because it can comprise the heterozygous population and 100% homozygous of dominant allele, 100% homozygous of recessive allele. So these 3 combinations should make up 1, Therefore, P plus Q equal to 1. And similarly, P equal to 1 minus Q. So, if we can just square it, like P plus Q whole square is equal to 1, then so P square plus 2PQ plus Q square will be equal to 1. So therefore P square is the frequency of the homozygous dominant population, that is AA, capital A, and 2PQ is the frequency of heterozygous or capital A small a people, and Q square is the frequency of homozygous recessive ones. So it can be denoted like this. So from observations of phenotypes, we can only understand the frequency of homozygous recessive or Q square. Those who express the trait in their phenotype, it could be either homozygous dominant or heterozygous dominant. So the Hardy-Weinberg equation allows us to predict which ones they are. Since P equal to 1 minus Q and Q is known, it is possible to calculate P as well. Knowing P and Q, then we can put them in this equation and we can understand what is the frequencies of all the three genotypes in the population. So this is Hardy-Weinberg equilibrium equation. So basically we can understand what is the genotype frequencies of all the three genotypes, like homozygous dominant, homozygous recessive and heterozygous. So coming to that, here are two examples given. Suppose this yellow phenotype, the number of individuals among 500 is 245, this is 210 and this is 45. So the genotype frequency is obviously calculated as the percentage. So this is like 245 out of 500 is 49%, and this is 42%, this is 9%, which is denoted in decimals. So therefore 49% is 0.49, and so number of alleles in the total pool. So 500 individuals will have 2 copies each of the same gene or same trait, and therefore the number of alleles will be 500 into 2, 1000. So among this 1000 alleles, capital Y will be 245 into 2, because the number of individuals is 245, each will have 2 copies. So 245 into 2, that is 490 plus 210. 700 capital Y will be there, and similarly if you calculate the small y, it will be 300. So these two make up 1000. Now we want to understand what is the allelic frequency or P. So P is 700 Y divided by 1000, that is 0.7. So 0.7 is P, and therefore P plus Q is 1, therefore 0.3 will be Q. So these two are known. Now P is known, Q is known. Now we want to understand what is the predicted frequency of this homozygous recessive, homozygous dominant, homozygous recessive and heterozygous. So how can we understand P square plus 2PQ plus Q square equal to 1? So we put the values of P and Q over here, and then we get this frequency which was given over here. So this is the Hardy-Weinberg principle. From this equation itself, we can understand what is the frequency of this. Now coming to this parent generation 1000 individuals. So from here also you can understand that 360, 480, 160 both make up 1000. Genotype, each genotype is 0.36. Same way if you calculate the percentage, it will come out like this. And from here, if we calculate the allelic frequency, it will be 0.6 and 0.4 P and Q. So from here again, if you give the equation, that will be P square plus 2PQ plus Q square, then it will come out like this only. So this is another example of Hardy-Weinberg principle. So the evolution types mechanism ends over here, and in the next video I will start with animal behavior. Thank you.